And the final speaker for today, this session, is Dr. Csaba Kerepesi. Did I pronounce it correctly?
Csaba Kerepesi.
Okay. He's going to talk about methylation and network entropy measurements during aging. Sounds interesting. And he's from the Unfried, Budapest, Hungary. Thank you.
Yes. So I'm Csaba Kerepesi from the Unfried, Budapest, Hungary. And nice to be here. And I want to talk about entropy and networks. So what is entropy? How is it related to aging? Entropy is a measure of disorder in the aging system in which death is the maximum disorder. So in my thinking, the Yong system is ordered and associates with low entropy, and how it is work. And the old system are disordered and associates with high entropy. And aging is the transition between young and old. It increases the transition between low entropy and high entropy at the same time. So this is the theory. And my main questions are, how can we measure entropy in different data types, maybe? And are entropy measurements suitable to evaluate accelerated, slowed aging or rejuvenation? What is the relationship between entropy measurements and aging clocks? So when I was in the Gladyshev lab, I was a postdoc for 2 years. I had a luck to analyze the naked more rat, this potential non-aging mammal. And actually, we found that it ages epigenetically, because we could develop an aging clock. We could predict the age of the naked more rat very accurately. You can see here the results. And but we also measured the methylation entropy, the mean, and found that the mean methylation entropy increased with age significantly after 1 month. So how can we measure methylation entropy? So methylation is normally 0 or 1, so it makes sense to have the minimum entropy for these extreme values, so 0 and 1, and the maximum entropy is at 0.5 methylation level. There is 0.5 methylation level because this is bug data. So, and actually I realized that the clock, the naked more rat clock, that contain only very few number of CPG sites, only 26. The increasing size you can see here starts from almost 0, and the decreasing clock sites start from almost 1. Sorry, this is 1.0. And actually these are, in these clock sites, entropy increasing. So these are maybe the clock, the algorithm selected the most, almost the most entropic CPG sites. So this is the relationship between epigenetic clocks, a close relationship between epigenetic clocks and entropy, I think. And in a recent study, we analyzed high-capacity runner rats and low-capacity runner rats. These are selectively bred rats after 44 generation. We have super fast and super slow rats, and in a collaboration with Zsolt Radek in the Hungarian Physical University, we measured 4 organs and RRBS, bisulfite sequencing methylation, and interestingly, we didn't find any changes between the groups by using any clocks that we could develop in any organs. So it was quite disappointing. But when we measured entropy, we found significant differences in heart and large intestine. And the high-capacity running rats showed lower mean methylation entropy compared to the low-capacity running rats. And if we consider that actually methylation entropy increases with age, based on the Morgan-Levin's red data set, we can interpret this decrease actually as a kind of biological age decrease, slowdown in aging. The question is whether we can interpret it as it is, and how it is possible that the clock does not work, but entropy works. So I think answer can be that the mean methylation entropy uses almost more than 1 million CpG sites, and the clocks uses several hundred CpG sites, so it's possible. By the way, it's an interesting result that the mean methylation entropy is the highest in the large intestine compared to the heart, hippocampus, and muscle. I'm also very interested in networks, so how networks ages. I think it's very interesting. And here we analyzed brain networks. We constructed brain networks from the braingraph.org website. We downloaded the data. These were constructed from diffusion MRI, from the OASIS 3 and Human Connectome Project data. And the nodes are the brain areas, and the edges are the connections between brain areas. We have one edge if we measure at least one axon fiber between the two areas. And there are weights. Weights are the number of fibers. So you can see here the brain graph, the brain network for the lowest stage. Let me consider 45 years young, as I'm exactly 45 years old, but it's not my brain graph. And this is the oldest guy with 95-year-old, and you can see some difference. Maybe the right one is disordered a little bit, lost some edges. But how can we measure it? We can count the number of edges, and you can see the number of edges decreases significantly with age. And we can measure entropy, but measuring entropy for networks is quite tricky. We implemented two types of entropy measurements. You can apply Shannon entropy for the degree distribution of the network, and we have another entropy measurement, the random walker Shannon entropy. So basically we consider random walker on the graph, and we calculate Shannon entropy for the probability distribution of the random walker. You can see here, you can check the full formula in Wikipedia as well. But interestingly, and somehow again, it was a disappointment, that entropy decreased with age. So I don't know why. I have more questions than answers in this talk. So maybe these measurements are not suitable to measure entropy. That could be an answer, or we could try other entropy measurements for networks, or we need to develop aging clocks for networks, maybe. That would be a new thing, because I think nobody used networks as an input for aging clocks. The other possibility to analyze network to investigate these very interesting questions, how networks ages. This is the protein-protein interaction network. We constructed it using single-cell RNA-seq data and string database, where we can find the protein-protein interaction network, the only one for human. And protein-protein interaction very important in the cell. So proteins are very important, of course, and the interaction between protein also very important, maybe the most important thing in the cell functioning. And it's, I think, very important to investigate how it changes with age, this network, these connections. And so we constructed networks for each cell, each single cell, by restricting the protein-protein interaction network to the nodes with non-zero gene expression. So we have different networks for each cell. And we could investigate how network properties correlate with age. We analyzed the tabular sapiens data sets and find a small decrease in the number of edges. And we calculated entropy measurements and found some constant or increasing changes. But Vadim mentioned in his talk that we found a rejuvenation event previously in his lab in embryonic development. And currently, with my doctoral student, we confirmed it in single-cell transcriptomics. So you can find a decrease in human embryonic development. Both data set confirmed it. So I was wondering whether the simply mean methylation entropy measurements recapitulate to the finding of the aging clocks. But the results quite controversial. So for the edges, we can see a decrease, but we would expect the opposite, an increase if we consider rejuvenation. And in entropy, again, we found the opposite effect, an increase in entropy. So again, a question. So my answer could be that maybe we need other entropy measurements for networks, or the clocks may be more accurate measurements of rejuvenation. This simple measurement that mean global methylation is not enough, or we need to develop, again, clocks for networks, this type of networks. And, this is my lab, my research group. I have a luck to have a lot of students now, and thanks to everybody, every collaborator, and the students, and special thanks to Vadim Gladyshev, my former supervisor and mentor. Thank you for listening.
Thank you.
Very nice talk. I was just wondering about the methylation-based entropy, tissue-specific. You saw that in large intestines it was high, and in the heart it was in the middle, and then in the hippocampus and muscle it was low. I'm wondering if this correlates with cell replication. So cells that are highly replicating, they may have a higher turnover of methylation loss. If this is something you've looked more into?
Yeah, that's a good point. Thank you. We need to check it, whether there's a correlation with cell replication and mean methylation entropy. Thanks.
Yes, thank you. The great talk about entropy. I love the concept of entropy, really. Not really a question, but rather a comment. There is a nice observation that entropy in high-dimensional physiological parameters, like, for example, heart rate variability, very known in aging, especially parameter, entropy actually decreases with age. But entropy of DNA methylation, for example, increases. Obviously, the entropy of DNA itself, in terms of somatic mutations and other different modifications, information entropy, also increases. And this is a very nice, peculiar thing that on the molecular level, the entropy increases with aging, but on the physiological level, entropy of different physiological signals decreases with aging. And this is just an observation. Maybe you can comment it, but it's just my comment.
Yeah, thank you. Very nice comment. Yeah, we need to investigate more different entropy measurements. So this talk was actually a thought-provoking talk. If you want to discuss entropy, just come to me, and I'm also very interested in how can we relate entropy and aging clocks and everything. Found out better entropy measurements.
Okay, thank you, Dr. Kerepesi, for an interesting talk. Okay.